So there we are going to go kind of quickly through this one. There's a lot of neat stuff I'm going to show in class, but I'm just going to hit the straight IB stuff here. It's either pretty close to Halloween or just after Halloween, so be careful on Halloween. So that one will do like at home. There's your EOC. Notice these are all in purple. That means this is higher level, but that means, again, most of you are taking higher level, or I know a good bit of you are. So that's what we're looking for here. All of it's purple, just a little bit of red, and a lot of the red has to do with these things that we're going to hit again in a unit on classification, and I've been throwing them in here or there because they're not very specific about recognition of which features are we talking about. So eyes here is a neat story, and so we're going to talk about eyes, and probably the only eye that might make their list of distinguishing between different kinds of things is the compound eye of these, which are arthropods, right? And that's the phylum name that is on their list. Arthro means joint, right? Arthropods have this compound eye, and that's probably the only thing they'll ask. We'll look at some neat stuff in class. We do have this idea that the gene that directs the development of eyes, which we bumped into before, that kind of gene called a Hox gene, is common to all animals, which is kind of neat because even this very different compound eye of a cockroach, for instance, has the same gene that orchestrates its development as yours and mine. That was a big discovery. So we're going to look at it right here in class, and then here a little bit of evolution stuff here that's kind of interesting. So we're just going to do this in class as a kind of a demo stuff, but you don't need to know anything about how eyes work. That is one of the optional things and a different option that we wouldn't have done anyway, even if we were in an optional world. So this is kind of neat about, here we go again, because we're mixing all kinds of things in here. This I put as an example of the fact that for a cell, here's the cell, that has some kind of receptor that can take a stimulus and turn it into some kind of action. In this case, an action potential. In this case, a nerve impulse, which we just got finished talking about. That stimulus doesn't have to be a chemical. It might be light. If regular ordinary light or some specific wavelength of light can make a molecule change in a way that triggers the dominoes to start falling, right? Which often involves a second messenger cyclic GMP in this case, but what it results in is sodium channels opening. So we're not turning on genes here, but it's the same kind of stimulus response with a little pathway, signal transduction, as the general idea of what that is all about. So we'll talk about that again just a little bit here. And hearing is kind of interesting. Here we have nerve fibers, right? In little parts of the inner ear, we have these nerve fibers that end in very hair-like, they're not hairs, but they're hair-like little extensions that wave back and forth when fluid sloshes around. In the chambers where those little cells are, here they're enlarged, right? The fluid sloshing around in these chambers, which involve this circular thing, which involve these semicircular things, semicircular canals. In different places in both of those, these nerve fibers will be triggered by bending this way or that way, depending on which way your head is bending, because they're up in your inner ear in your head. So we'll talk about that a little bit, and we'll talk about this a little bit, which is kind of interesting. Okay, so now the red stuff. Most of this is skeletal, how skeleton stuff works, and then they have this very specific stuff. They're going to want you to be able to label this. The next slide is going to have a better label with the things they want you to label. But here we have the fact that your bones are attached to anchorage muscles. Your bones act as levers then. When the muscles contract, it pulls on the bones and one of your body parts moves, right? Like your forearm. Same in a cockroach or a grasshopper here, except that they're attached to their exoskeleton because they don't have that inside bones endoskeleton, right? And then we have the idea that these muscles are almost always paired with a muscle that causes the opposite kind of movement, and that is antagonistic. There's that word. We had it with hormones, right? Application, antagonistic pairs of muscles in an insect leg. I don't know what it means, to be honest, when they say application, but to me it's like a threat. We might throw this kind of diagram in a somewhere in a question and either ask you to explain what it means or whatever. Just while we're at it, okay, if you've had anatomy and physiology, you remember these maybe. Flexion means you're going to bring these two closer together. Extension means you're going to push them further apart. So these antagonistic muscles, one of them is a flexor and one of them is an extensor. And for the ones in your elbow, which they might ask you to diagram, and this is not uncommon at all, these are the things they want you to be able to label. So the named bones, they didn't put the names right here, but they want those named bones. That would be your upper arm bone humerus. Nothing funny about that. Radius and ulna, you have to get those straight, right? The ulna is the one where you're like that most of your elbow, or actually all of that elbow bone is ulna. So however you want to remember them, the radius is the smaller one, the ulna is the bigger one, however, whatever. Okay. And named antagonistic muscles, which is simply bicep and tricep. Those you're pretty familiar with, I think. But this would also, if we put them with that other, on the objective on the last page, can't spit that word out, right? When the bicep contracts, it pulls on here and pulls your forearm closer to your upper arm. That would be the flexor, right? This would be the extensor. When this contracts, it pulls on this side of it to swing it that way, extension. Okay, so so these other things, notice you have joint capsule, right? Capsule is like an enclosed, it's almost like a balloon that's squashed. In fact, it's quite like a balloon with tougher walls than your normal balloon that's squashed. That's the capsule part of it. Inside, there's synovial fluid. They wanted you to do that. And then you also have some pads of that kind of connective tissue called cartilage, which are doing what I just said. They pad, they cushion. In a knee joint, for instance, you might have know this word. That cartilage pad is called meniscus. So anyway, they didn't put that specific word in there. All right, so now lots of words here, lots of specific diagrams, other diagrams that they want you to do, even to analyze an electron micrograph to say whether this muscle fiber looks contracted or relaxed. That's what they mean by that, the state of contraction to muscle fibers. Okay, sorry, but we'll go back this way. And in the next little video that we'll see in class, this is kind of spelled out in video style. But here's the words they want you to talk about. Muscle fiber and muscle cell are synonyms, right? They use the word fiber here, but it is synonymous with cell. These are cells that have many nuclei. You can see that here in the diagram, right? This is one muscle cell. What a muscle is a bundle of these muscle cells. And what a muscle cell is, this muscle fiber, is a bundle of myofibrils. ILS on the end of that means little, myos the muscle prefix. So what we have here are bundles of bundles of bundles, literally, right? Because each of these myofibrils is itself a bundle of these smallest little cellular components called microfilaments, and they're of two different kinds. The myosin kind, which are a little thicker, than the actin kind, which are a little thinner, and they are typically called thin and thick filaments, as well as their more biology name. Okay, so now if we look at this sarcomere, remember this, this myofibril is just inside of a cell, a muscle cell, right? And there's a bundle of them, almost like you had a handful of pencils, and each pencil was a myofibril. And the whole handful of pencils is the muscle cell. That makes sense. Surrounded by a plasma membrane, all that stuff. Lots of nuclei kind of squished to the edges by all of these myofibrils inside. Well, each myofibril is a series of sarcomeres. sarco is also kind of a muscle word, right? And this sarcomere, they want you to be able to identify a labeled diagram, and they tell you what they want you to include. They didn't include all the stuff in there, like this H zone and I band and A band and M line, right? But they did include Z lines. The sarcomere is defined basically by Z lines. That's a Z line, and a sarcomere is from one Z line to the next. So they kind of define the outer edges of each of these units called a sarcomere. And the Z line of this one is also a Z line for the one that's over here, right? They kind of share Z lines, so to speak. The actin filaments, we're going to see all this in animation, a really good animation. The actin filaments you can see from here are attached, physically attached to the Z line. All of these are protein molecules, right? And then the myosin filaments have little heads, which we need an expansion for. And these heads are going to attach to, and see they said that, these heads are going to attach to the actins in a attach, disattach, attach way. And this is how they're going to move to cause that whole cell to become shorter and thicker, contract. And then here you have, because they want you to like be able to diagram this, but more likely they want you to describe what happens to these light and dark bands when this muscle contracts and when it doesn't, because a muscle contraction is due to each individual sarcomere contracting and relaxing. So what makes these bands light and dark is basically where the myosins are interacting with the actins, where you have both myosin and actin in the same place, that makes them look dark, right? If you only have a place where there's only actins on either side of the Z line, that makes them look light. What we're going to see is in a contracted muscle cell, the light space kind of disappears as these Z lines get pulled closer to each other. The light space kind of disappears and the Z line is like right up next to the myosin. I know it's getting messy. Okay. So they might even ask you with one of these to measure the length of a sarcomere with all the stuff we'll do when we get to microscopes, a little bit of magnification math. Okay. So here, how does this work? What we just looked at was the anatomy of it, right? We named all the parts, and that's what anatomy is. And now we're looking at the physiology part of it. How do these actins and myosins interact in a way that causes this sarcomere unit to get shorter and thicker? Therefore, each myofibril, therefore, each muscle cell, muscle fiber, getting shorter and thicker, right? We have ATP hydrolysis happening. We have cross bridges. These again are words in the syllabus. You need to have them in your vocabulary, right? Sliding. This model is referred to as the sliding filament model. All right, and calcium ions play a role, and these two proteins, troponin and tropomyosin. So we're going to look at all of that stuff when we look at this video coming up. Okay, so on the next page we'll see that video, but I probably won't watch it here. We'll just talk about it here. So now what we have in this picture is just one little part of what, again, a sarcomere has a bunch of. Here's one Z line, here's another. Here's a whole bunch of myosins. Here they just have one myosin right here, right? And over here is a whole bunch of myosins. And in between, attached to the Z line, get out of the way, attached to the Z line, and in between each myosin are the actins. So they have taken just like one of these units with one myosin and an actin on either side of it. And if you can, in your mind, think 3 dimensions. There are other myosins around it, like surrounding it in 3 dimensions. It's not 2-dimensional. Anyway, so now they really close up the actin, thin filament, right? The myosin, thick filament. Remember, all this is proteins, fibrous proteins, and a part of that protein sticks out here. is referred to as these myosin heads, and that's the part that is going to attach to these thin filaments and move them along. This is very much like the motor proteins that on microtubules can move along microtubules by attaching to them, disattaching, attaching, changing their shape, disattaching, changing their shape. They are walking along here very, very fast, but they're walking. What's going to power the shape change is ATP. Okay, so when ATP attaches, you can see here ATP attaches, that causes this to attach. It grabs the myosin in a place where we're coming up to that troponin, tropomyosin part in a second. That's a little more detail, right? And then it changes shape. It changes shape back to here. It like pulls it that way. We'll see this better on the next. And then it lets go. It flips back to this and grabs again. So it's like a person grabbing a rope and pulling the rope and then letting go with that hand, moving it to another place on the rope, and hand over hand pulling that rope towards it, moving that rope across the myosin head. The animation's really going to help here, okay? So now the tropomyosin and troponin complex part. So you see the tropomyosin is a molecule that's surrounding the actin filaments. And the actin filaments is made up of units that have a place, this little kind of brown circle place you can see down here and up here. That is where the myosin arm can bond to. Myosin head, sorry, they call it heads, right? My analogy was like it's your hand grabbing a rope. Well, this is where they grab the rope. This is the rope they grab, the actin, this is where they grab it. But in a relaxed muscle, that place where they grab it, the actin attachment spot is covered up by tropomyosin. Here's what happens when you let calcium into this muscle cell. The muscle cell has a bunch of calcium in its endoplasmic reticulum. It's just waiting for a nerve impulse to say, hey, open some calcium gates, spill this calcium out over this thing, and if the calcium gets spilled out over this thing, then what also is going to happen, because the calcium's attached to the troponin, is it's going to change their shape and it'll pull that tropomyosin out of the way. Now those myosin heads can bind to the myosin binding sites because now the tropomyosin is out of the way. You really have to practice looking at this for a while, and so that you don't mix up the very similarly named tropomyosin and troponin, right? And their thing here. They're kind of part of the same protein complex. The tropomyosin is the long thread, and the troponin is a little more globular complex. And what you have to remember is that's the part that the calcium is going to attach to, the troponin. And when it does, it causes the troponin to change shape and pull that thread-like tropomyosin out of the way. You'll see all that in the video. If you're in class, you already have seen it on the video. Okay, so now we look at a little bigger picture. This did not make the syllabus, but I'll just throw it in there just as an example of cell parts and specialized cell parts. Here you have that end of an axon branch from the kind of a neuron that's going to make something happen called a motor neuron. And this then is a neuromuscular junction, the same as a nerve synapse, but we give it a different name if it's not connecting to another nerve, right? We have acetylcholine being given off. That you saw that all on the video if this is in class. But what that's going to do is open these calcium gates. When that neurotransmitter acetylcholine hits the receptor here, signal transduction pathway, it's going to cause the calcium gates in this endoplasmic reticulum, specialized in a muscle cell, that's called sarcoplasmic, but it's still endoplasmic reticulum, to open those calcium gates and spill the calciums all over the the sarcomeres, right? The myofibrils. Here again, just got to keep looking at this over and over to not get myofibril and muscle fiber mixed up. This is a whole muscle fiber, right? It's got a whole bunch of myofibrils in it, like a bunch of pencils, like I said, held in your hand. And in between here is a lot of mitochondria cranking out those ATPs, so on and so forth. Okay, so this is going to be fun. They suggest that we do this. You know, they don't test you on this, but this is a good little lab that easy enough for us, because now we're getting into the muscle fatigue part, which we talked about before, way back when we talked about what if you can't get enough oxygen to this muscle fast enough to have it work to the demands that you're having it work, which means you're asking it to work too much, and that consequent lack of that muscle working is what we call fatigue, right? It's not able to contract as much as it used to be able to. We're going to use these little grip strength things to have a contest real fast. And there's the different kinds of muscle, which again, they didn't need for you to know. There we go. Hope you liked it.